Today's video is focusing on what I wrote for my personal statement. Hi guys, welcome back to my channel, Seun in Science. I'm Seun and I'm in science. Okay, so now let's move on to my most recent experience before I started my PhD. I've titled this Ongoing Experience, and this is now talking about my time as a research assistant, which is in my current lab still. As a research assistant, I've undertaken 2 projects, including performing literature searches, experimental design and setup, results interpretation, and oral presentations. My initial project explored the function of MBD3 NERD in human-induced pluripotent stem cells to determine whether NERD is required for lineage commitment of human-induced pluripotent stem cells. Wild-type knockout and rescued human-induced pluripotent stem cells were induced to differentiate towards a neuroectodermal or definitive endoderm fate, and dynamic expression changes were analyzed via RT-qPCR. In addition, I utilized immunofluorescence to see if the cells are responsive to the directed differentiation protocols. Culturing these in a lab that works solely on mouse embryonic stem cells granted me the opportunity to simultaneously demonstrate independence on the project as well as reach out and learn from other labs in a collaborative nature. My contribution to the project is included in a recently published paper in stem cell research, and again I've given a link. Furthermore, the chance to present my project as a poster during the annual stem cell retreat in 2019 strengthened my competence in scientific communication and discourse. So just in the first paragraph, like, there's quite a lot of information jam-packed in there. One thing that I've done in the first few lines is give literally an overview of some of the key technical skills that I've learned. Not technical in the sense of, oh, these are the protocols that I've done, but actually what are the fundamental research techniques and skill sets I've learned. Number one, obviously reading papers, right? You're reading papers, consolidating the literature, trying to get information from here and there, right? Designing experiments in the most optimal way. Of course, there's going to be troubleshooting that is underlying that. Interpreting results, right? Not just that, okay, I do an experiment, get results, and then I pass it off to someone, but I'm actually there for the whole process, the whole scientific process, as well as then presenting my data to people. And this is kind of encompassing the fact that I present it to lab meetings, but then at the end I tie that back in, re-emphasizing on this aspect of oral presentation by demonstrating that I've presented it to a wider audience. And again, with what I've mentioned earlier, I've given, I've given an example, I've given a blurb of what the aim of the project is, what am I exploring, and I've given an idea of how I've gone about doing this. I've taken these cells, wild-type knockout and rescue, I've done some differentiation protocols with them, and I've used different techniques to come to a potentially similar result, right? I've done the differentiated protocol, I've done gene expression analysis using RT-qPCR, and then I've done some staining, I've done some immunofluorescence to confirm what it is that I would have seen by gene expression. So again, it's this kind of re-emphasis of I'm able to use orthogonal approaches to come to the same results and reach and establish the same conclusion. But again, I'm showing this. I'm not just telling that, oh, I know this and listen things off, but I'm giving an insight to how I've gone about developing these skills, how I've gone about interacting with my project, interacting with my data. More recently, I'm involved in investigating how chromatin remodelers, CH4 and BRG1, act to dynamically alter chromatin structure, enabling pluripotent cells to change the transcriptional state and therefore respond to extracellular signals. As part of a team, we are investigating this by observing how inhibition of cell signaling affects the expression of ERK-responsive genes in cell lines where the remodelers can be depleted with a fine temporal resolution in mouse ES cells. Now, to be fair, this is a bit of a mouthful, but it does give, again, an overview of what my current project of what my most recent project was, right? So I'm now highlighting that, okay, I'm working with fine temporal system, I'm working in mouse ES cells, I'm working as part of a team, and I'm doing this in a collaborative way. To this end, I have generated, so this is something to highlight, that it's good for you to have strike a balance of what have you done as a person, as an independent unit, but how also have you contributed to a team effort. So in this paragraph alone, I have started off talking about how, you know, as part of a team, I'm contributing to this project, and then I am now highlighting, okay, what are the roles that I myself have played in contributing to this project. So to this end, I generated cell lines using the CRISPR-Cas9 technology. I've characterized and validated these cell lines via genotyping and Western blotting. So again, what I'm doing, I'm hitting 2 points here, and number one, showcasing what I have done in my time in this project, but also giving that kind of follow through of experimental design, that okay, I want to make a cell line, but of course if I make the cell line using this specific technique, I also have the voice of reason and the scientific knowledge to validate the cell line. But then I'm not just validating it using one technique, I'm again using orthogonal techniques. So for this, I'm highlighting that I'm aware of the different approaches that you can use to reach a combined outcome. But then by using the different approaches, you're really like putting more power in your side to show and to kind of hit the nail on the head that okay, I have not just this one evidence, I have this other evidence and this other evidence to really strengthen my point. Okay, so again, describe what I have done. So I've carried out time course experiments to look at the dynamics of gene expressions. So again, now I go on to explain what I've done specifically. Cells were harvested for RNA extraction for analysis by RT-qPCR. We wanted to assess the impact of loss of either protein on the nucleosome structure and chromatin association of transcript factors. So I learned how to perform MNAs and CHIP. Right, so I like this because, you know, it's showing that okay, again, I'm describing what I've done specifically in the project, but then also showing that okay, well, we had this need, so that means I had to learn this. I took the initiative to learn this because that would contribute and that would push the project further, because then you have more than one person who is adept, who knows these skills, who is becoming slowly and surely an expert in this aspect. As a result of my work, we now have chip-seq data that is being analyzed by our bioinformatician. So by again this statement, I'm showing and I'm trying to reflect that just the intricacies in my interaction with people in the lab, that not only do I interact with those in the wet lab, but also the data that I'm generating is not just ending off somewhere and not going further, but actually is being processed by other people in the lab. Overall, my time here has developed my ability to manage multiple projects. Other experiments have yielded opportunities to culture rat ES cells and troubleshoot converting mouse ES cells to formative cells. I haven't established a human culture system. The lab's work could move in a more translation direction in the future, and I'll have the opportunity to train others in these techniques. Again, I think this is a very strong end, showing that okay, again, I've worked in the lab, I have contributed to the team, I have worked independently, I've shown initiative, but also some of the skill sets that I have done independently, because again, the lab didn't work with human cells. I'm now in the position that I can push forward those knowledge onto those in my lab. I can train others in my lab, and this is highlighting that you have competence, that you know you're able to have gone into a lab being the one who needed to be trained, and now you're now well established in the lab, so much so that you're the one training someone. Okay, so this has just been an overview of me talking about my experiences so far, and as I mentioned before, these are going into so much detail, and really that provides you the opportunity to really reflect what are the main things that you've learned. Now I think if I just take a step back, some of the things that I think you want to try and get across is, number one, okay, describe in detail what was your project on. Right, what was the aim of the project? At what point did you enter the lab? What were the biggest questions that we wanted to address? And then what were the objectives of your project, right? How does it tie into where the lab was? How did you go about learning and addressing these questions, right? But I think the biggest thing that you really want to emphasize on with a lot of this is, what are the things you've learned, right? Reflect upon your experiences, right? You want to show that, okay, during my time there, I've learned a whole scientific kind of circle of nature. Is that the right word, right? Just kind of that thing of, okay, I have a question. How do I go about approaching this question? Okay, I have this experimental setup. Cool, I can reflect that in my writing, right? I know how to then go about addressing these questions. I know how to obtain results, but on top of obtaining results, I can interpret it in other ways, in different perspectives, from different kind of viewpoints. right? And then on top of that, what are the skill sets I've learned? And in addition to the technical things that I would have learned, I've learned communication, I've learned teamwork, I've learned how to be independent, I've learned how to organize my time, I've learned how to present my data to people, I've learned how to be critical about my data, like within myself, in conversations with people. I've learned how to talk about research on a bigger scale with those that are either within my field or outside of my field, right? I've been able to demonstrate that I have been trained, I've become competent in this skill, and I'm now in a position that I can share my knowledge with others. I've taken initiatives in different things where I've seen this gap in the lab, gap in knowledge in the lab, and I'm able to bring together some sort of like maybe background expertise or background knowledge, and develop on that so I can contribute effectively to the lab. These are the things that you really want to try and show and present and demonstrate in this section. Then the next section was I guess the personal statement aspect, or the statement of purpose. And here the prompt we got for this was, please summarize your research interests, career objectives, and the reasons why you've applied to study for a PhD and why you've applied to the Crick. Now, my one thing with this is, always respond to the prompt. They've given you the prompt for a reason. They've literally told you the things that they want you to tell them, right? So that's already the biggest hurdle for you to try and address. When I was writing this, I literally broke this down into sections. So I put on here, okay, what are my research interests? I'm gonna put a paragraph to that. What are my career objectives? What do I want to do after my research, right? Why have I applied for a PhD? I think for me, that's like, for a lot of people, that's a very difficult question to address. I mean, that's something that comes up in interviews as well. And again, I've got a video coming up that will kind of talk through my experience and show you in real time my PhD experience and when I was being interviewed. But I think it's a very difficult question for a lot of people to answer, but it really has to be quite specific to you. And I know that's easier said than done, but you know, it's better than being like, oh, I want to do a PhD because I want to be this, because frankly, there might be other approaches, other ways that you can reach this career objective potentially without a PhD. So you really have to go like really intrinsic and think, okay, why do I want to do a PhD? Why is it different from me being an RA or just, I don't know, doing a master's and jumping ship? And then specifically they said, okay, why have you applied to the Crick? Why the Crick, right? And this is where you really want to big up the name, like, okay, y'all are the best of the best of the best and I want to be here, all that kind of stuff. Now I want to talk about, you know, what I have written and how I have addressed some of these prompts. So I have just gone straight in the deep end. Like, I've already spent the other sections kind of establishing who I am, establishing a little bit about, you know, my experiences. So I don't have to be saying, oh, you know, I did a undergrad in this or a master's in this, because they will already say that from other aspects of my PhD application, right? So I've just gone straight into the deep end, addressing the first prompt, my research interest. So I say here, my interests lie in cell and developmental biology. Boom. Right? And this makes sense because the labs that I chose in the application round also align with that. So it's almost like, okay, you're doing like a click and sort thing. Okay, you're interested in cell and developmental biology. The labs that you've selected are also in this field. Okay, that makes sense. There is synergy there. You're not confused, right? You've already ticked the box. And then I've gone into detail about, okay, what about this field fascinates me? What makes me passionate about wanting to devote the next 3-4 years in this kind of niche field, right? So I've said here, I am fascinated at how information embedded within a single genome can generate the immense cellular diversity necessary to establish an organism. In particular, the unique potential of pluripotent cells to give rise to any cell type in the body makes them central in addressing a wide range of exciting scientific questions from basic and translational perspectives. And the reason I went for this angle was because of the labs that I chose. I will talk in another section which labs I selected at this round, right? So here was me showing that, okay, yes, I've selected labs that focus very much on basic, and by basic I mean, like, you know, fundamental biology, not basic as in like you can't sit with us, but basic as in, like, you know, it's not something that the most translational has the most translational objectives. But also I'm showing that, okay, I have that interest not just from, okay, I want to do basic research, but also at some point would want to take on to translational aspects and actually do have evidence of working on something that you know, to some level would have had a translational perspective. And I say that, and I mentioned that here. I have previously done fundamental research on human-induced pluripotent stem cells, but I am keen to transition to more translational studies, for example, using these cells to understand cellular decisions in making disease models. I am also interested in the mechanisms of self-fate determination using 3D culture systems such as organoids to model development. And so literally in this paragraph, I've talked about all of my research interests, and I kind of given like a blur, but I've made them almost seemingly transition, right? And really the underlying and the underlying structure to this that made the most sense was to go from very basic to, okay, what my ideas are of working in this field would be from a translational perspective. Right? And I've said here, reflecting on my past experience. So now my next paragraph really is to kind of address this bit. This is how I've decided to do it. I decided to address this aspect of why do I want to apply for a PhD. Okay, so I've said here, reflecting on my past experiences, I have learned the demanding nature of lab work. It requires careful planning, attention to detail, and an ability to look at results from multiple aspects for interpretation. And really and truly, the stuff that I've talked about in the previous sections does confirm that. I have shown, not just told, I have shown all of these things. The repetition of experiments or generation of negative results can be frustrating. Despite this, I enjoy research. And when I had this in, I had people looking at it, and you know, it was a bit 50-50 on whether or not I should input this, because you know, sometimes when you write personal statements, you want to kind of show this overtly like positive outlook on research, but I think it's more important for you to show that you have a realistic outlook on research. I had spent enough time in labs and done enough kind of projects to understand that research science can be frustrating sometimes. There was a period of time where, like, you know, for months I just had negative data, like nothing worked, and it was almost depressing. But the fact that I have that experience and I have that awareness of that, you know, literally science can be like that. I think presenting that shows that, okay, you really are aware of the ups and downs of a PhD, right? You're not going there all doe-eyed and like, oh, I'm gonna like save the world and cure cancer, right? So I think for me, adding this was kind of to highlight that I have a realistic outlook on what research is. But then I'm like, okay, despite the fact that it's not you know, all nice and dandy every time, I still enjoy research. I still enjoy, you know, the aspect of thinking critically, of trying to optimize the data. And for me, I feel like I didn't even need to kind of make this statement of, oh, I enjoy research because I still optimize data, because again, my description of my previous experience should highlight that, has highlighted that. The opportunity to gain leadership of my work, to be intellectually challenged, to drive experiments that address questions that I'm really interested in, and the thrill of making a novel discovery are my motivations to pursue a PhD. Boom. Right? Not just one reason. I have several reasons for why I want to do a PhD. And I think for me, it was important for me to communicate this, because I knew that a potential question that I would get personally, as someone who has worked in Cambridge, or someone who has already stayed in science for a while before the application as a research assistant, you know, it'll be a question of like, okay, why not just stay as a research assistant? For me, the reason I wanted to do a PhD was that I would have the opportunity to chase the questions that I am interested in, because as an RA, you know, you're never really like addressing questions that you're interested in. Your name is never going to be, you know, stamped on it. Yes, you'll be in the papers, but you're never going to be first author. You're never going to be, you know, having the opportunity fully to chase something or to explore a side project that it's fully yours, right? You don't have that full independence. Now, as I say, doing a PhD, I mean, I'm going to do a video on this. Doing a PhD compared to being an RA, it is like cats and dogs, completely different, and the transition was quite difficult, really and truly. But for me, you know, reminding myself of that is like, okay, yes, these are my motivations for doing a PhD. I don't want to just be, you know, someone I get told what to do and I do it and like boom. I want to have to like be in a position where I'm thinking more creatively. Okay, there's this question, there's this gap in the knowledge, how can we address this? You know, what does my data say? Well, how does it motivate, how does it direct the questions I ask next? Right? So now my next paragraph is to now address this part of why have I applied to the Crick. right? So what I have said is the range of high quality research, especially within development at Francis Crick, is attractive, and I believe the nurturing academic environment and strong collaborative nature will fulfill my aspiration to become a scientist. The opportunity to exchange ideas with peers and distinguished academics across the partnering universities will push me to critically evaluate my scientific ideas and ultimately propel me to become a scientist with important accomplishments in the future. Now, what I have done in this paragraph is actually hit two different things. I have hit two different prompts. I've talked about, why I'm applying to the Crick, and also reinstated or emphasized on what my career objectives are. So in the first part of the paragraph, I've talked about, okay, what are the things that is offered at Crick that is attractive to me, right? I've talked about the range of high quality research, especially in the field that I am applying to. I think that's something that is important to emphasize, that okay, I'm not just applying to you because maybe you're the top in the world, but actually you are the top in the field that I'm interested in, and that's why I'm coming to you, right? Then I've talked about, key things that it's part of the way the program is designed. So again, I'm sure you would know, I hope you know this, is that with Francis Crick, they are actually an independent institution. So they're not like an academic institution, right? So they are collaborating across with King's, UCL and Imperial. So once you are put onto the course, then you need to sign up to those other ones, right? So me then saying, okay, the strong collaborative nature will fulfill my aspiration to become a scientist, I'm making note of that, and I re-emphasize that here where I say the opportunity to exchange ideas with peers and distinguished academics across the partnering universities, right? Again, highlighting that I have read your webpage, I know the design of this program, right? And I like the design of this program. And then I ended off with this. So while science is indisputably my vocation, I believe that promoting public awareness and appreciation for research is part of our responsibilities as scientists. I am particularly passionate about addressing the issue of diversity and representation in STEM. I have spearheaded an initiative to share my experience as a BME personality on digital platforms such as YouTube, Instagram and Twitter, and this has recently culminated in receiving the Institute's Public Engagement Prize. Continuing these endeavours during my studies at Francis Crick will enhance my proficiency in communicating scientific concepts to a non-scientific audience. While I hope my past experiences and achievements demonstrate my academic potential and tenacity, I am confident that my skills and commitment to research will contribute to the thriving scientific community at Crick. Okay, so here my last bit now is kind of to give another dimension to who I am. So yes, the focus of this is to touch on, okay, what are your academic interests, right? What are your research interests? But I really wanted to end this off to be like, okay, yes, in addition to me wanting to aspire to be a scientist, I have other aligning goals. I have other things that are slightly outside and can be put in parallel to my research that I'm passionate about, right? So it shows that I am more than just a scientist. I am a three-dimensional person. There are things that I do in my spare time. The things that I believe are, of good cause that I do in my spare time, different to what I do at work, right? So I think it shows that you have dimensionality to your personality. Of course, I have this platform where, again, share my experience as a black girl in science. I thought to mention that. I thought that would be the icing on the cake, right? And I then tied that in saying that, okay, these are things that I've done here in Cambridge, but it's something that I plan to do, and I know that the skill sets I gain from this will not just be beneficial to me as a person, but will be beneficial to me in my career as a scientist, because the ability for you to be able to communicate your science not just to fellow colleagues and fellow scientists and experts of your field is more important, and is and it goes along further when you can communicate those information to a non-scientific audience, to a lay audience, right? And then of course I've ended here with saying, while I hope my past experiences and achievements demonstrate my academic potential and tenacity, I am confident that my skills and commitment to research will contribute to the thriving scientific community at Francis Crick. So I always like to end on that kind of yes, I need you. Yes, I want to be in your place. But let me just remind you who I am. Let me just remind you that, yes, I literally do have the academic capacity. I have the relevant experiences, and I will be an asset to your community. So I think that's a nice way to end, to just end strongly and very confidently, and okay, yes, if anything, we both need each other here. So yeah, this has been my my own my own personal statement and talk about and rehash of my past experiences. Let me know if you have any questions, and yeah, good luck for application this season. Remember that I think the 10th of November, and then I think I'm gonna have like a series for Francis Crick. So I'm gonna have a separate bit where I talk about, you know, the interview round, because one thing to point out is that if you are called back for interview, there's actually like a pre-interview. I'm gonna leave it there, so y'all should wait for the next video so I give you more information about what this pre-interview is. Anyway, thank you so much for watching this video. If you have any questions, put them down below. Don't forget to like, comment, subscribe, and, you know, show your girls some love, and I'll see you in the next one. Bye.